Hey everybody, it's Mr. Hallahan. I'm going to be going over oxidative phosphorylation, and hopefully this will be a good companion to the reading that you did last night and your understanding of what's going on. But keep in mind, we will be going over this more next week. So do your best notes today as I go through this, and know that we will be going over this more next week. First thing we're going to do with this is we are going to be labeling where the different parts are, where we are in the mitochondria when we do oxidative phosphorylation. So first thing, this orange section right here at the top, this is going to be your intermembrane space. Next, this section right here should look familiar because it looks like something else that we've discussed this year. If you said cell membrane slash phospholipid bilayer, that's correct. In this case, though, we're calling it the inner mitochondrial membrane. And then lastly, the portion at the bottom is what is called the mitochondrial matrix. So we have all these different parts of where we are, but we have to put this in a context of where in the mitochondria are we. So in the book, they gave you a picture of a mitochondria. I'm going to do my best to replicate it. It kind of looks like a bean. So we'll do that. And then what happened was that bean had a bunch of folds on the inside, something like that. Okay. So what we're going to do is we're going to zoom in and show where we're at. So if I take one of these folds and draw a little square right there, what we've done is we are zooming in, and this section is the intermembrane space. The folded line is your inner mitochondrial membrane, and then lastly, the large section in the middle is the matrix. So one thing that we have to be sure we understand is why are there two membranes and what is the purpose of the folding, right? So the folding actually increases surface area. And as we discuss in biology, structure and function work closely together. And this is one of those times where it works together in trying to increase surface area. When we discussed cell size, we always talked about how a high surface area to volume ratio is good for the cell, allows for movement of molecules in and out efficiently. In this case, the folds increase surface area to allow the mitochondria to do as much oxidative phosphorylation as it can. The more surface area, the more oxidative phosphorylation, and therefore the more ATP that can be produced. Okay, so let's now get to the parts of the molecules involved. We're going to start down here on the bottom left, and the first thing we're labeling is NADH in that rectangle right there. Next, we have FADH2 right there. These two molecules you should remember from glycolysis. They have a very important job, and that important job is to be an electron carrier. So I'm going to do my best to show those electrons that will be present on those two molecules. So those electrons that are being carried by NADH and FADH2 were taken off of glucose and other molecules that were that glucose was made into, and now those electrons are being brought here. Now, as we look at this, we can see what's happening. I'm going to highlight in a different color. This yellow arrow that's now turning green with the highlighter is showing the path of the electrons that are being dropped off. So what you may notice is the electron carriers bring the electrons to the mitochondrial matrix, and then the electrons follow the path through the inner membrane into the intermembrane space, kind of, on this carrier protein, and then back down. Okay? So why does this all happen? Well, what you may notice is when those electrons are moving, H plus ions are also moving. So what you can see is these carrier proteins, which again, their names are not important, but I'm going to label them anyway because the book labels them. Realize though that you will not be held accountable to remember their names. So that one is 2, 3. We have site C, and then lastly 4. All right. So these are your carrier proteins. What are they carrying? They're carrying electrons, right? And what happens is they carry the electrons. When they carry the electrons, they use the energy electrons of the electrons to transport H plus ions to create what we called in our previous unit a concentration gradient. Okay, so we're basically concentrating all those H plus ions up here in the intermembrane space. I can even add some more up here. All right, so now NADH, it dropped off an electron, right? If it dropped it off, it lost it. And we know if you lose electrons, you're going through oxidative phosphorylation. So we're going to say that NADH turned into NAD plus and FADH2 turned into FAD. So the electron transport chain is what we just discussed. So we're going to label it down here at the bottom next to number 1. And that occurs from the NADH all the way to this point right here where the electrons are dropped off. Now, the electrons have gone through the electron transport chain. They're moving through these proteins, but then they have to go somewhere at the end. And where they're going to go is to the most electronegative option. Each time they were transferred from protein to protein, the purple proteins right here, they had gone from an electronegative protein to a more electronegative protein. What we're going to do now is we're going to talk about it goes to the most electronegative thing, and that is a molecule of O2. So that's why O2 comes into play now. O2 is going to accept the electron at the very end. That's why we call O2 the final electron acceptor. Now, when it accepts O2, when O2 accepts the electron, it also is going to combine with H plus ions that are floating around in the matrix. If you combine H with O2, you're going to get H2O. So to keep our counting of atoms accurate, it's going to be an H plus ion, 2 of them, plus half of an O2 to form H2O, just to make this balanced reaction. So we have gone through our process of electron transport chain. The purpose of that was to take the energy of those electrons from NADH and FADH2 to transport H plus ions into the intermembrane space. So what's the purpose of that? Well, if you create that gradient of H plus ions in the intermembrane space, they're going to then be able to move due to the properties of just diffusion. Those H plus ions on the top, they're going to be going through this molecule right here. This protein right here. And that protein we're going to label as ATP synthase. So should be fairly obvious what this protein is going to do, right? Given the name, synthase would imply that it's going to synthesize. And what is it synthesizing? ATP. That's why we have an ATP molecule right here. We discussed yesterday, when ATP is made, the reactants to form it is ADP plus P. Okay, so we now have a reaction where we took adenosine diphosphate and turned it into adenosine triphosphate, right? We learned yesterday what those prefixes mean. It's really important that we take our time and understand what those prefixes mean. All right, so we have now formed ATP, and that's through the process of chemiosmosis. Now, osmosis should sound familiar because we learned about that as being the movement of water from an area of high concentration to an area of low concentration. In this case, because it's chemiosmosis, it's not the movement of water, but it's the movement of H plus ions from an area of high concentration to an area of low concentration. Together, both of these processes are what make up oxidative phosphorylation. Now, yesterday we had mentioned how glycolysis, pyruvate oxidation, and citric acid cycle, together they form a very small amount of ATP, 4 in total. And then oxidative phosphorylation was going to produce most of the ATP. I mistakenly told you that it was 32. I apologize. In total, it's not in total, it is 32, but this process right here is making 26 to 28. So the 32 comes in if you combine the 28 of oxidative phosphorylation with the 4 produced in the previous processes. Okay, so just to kind of recap everything, oxidative phosphorylation is going to be your main producer of ATP. In order for this to happen, it needs to have the electron carriers coming over from glycolysis, pyruvate oxidation, and citric acid cycle. Those electron carriers are going to drop off their electrons, and when they do, those electrons go through the electron transport chain. When they go through the electron transport chain, the energy of the electrons is going to be used to transport H plus ions. That's going to be necessary because we want to create a concentration gradient of H plus ions. Now, when the electrons are done, they have to be accepted by O2 at the end. That's why it's our final electron acceptor. One key note about this is if there is not oxygen present, then the electrons have no place to go and they start backing up. It's kind of like cars on a highway when there's no exit and everything is stopped, right? Things are going to back up, back up, back up. You might be thinking back to when we discussed this on the very first day. When does oxygen not make its way into a mitochondria? Well, if you are doing very vigorous exercise and your breathing rate and your blood flow cannot get oxygen to your muscles quick enough, then your mitochondria are not going to have O2. It's not going to be there to accept the electrons, and then this whole process is going to back up. So you can imagine all these electrons will be backed up along the chain. If that occurs, the electron transport chain stops. If that stops, then chemiosmosis can't happen. And therefore, you're not producing 26 to 28 ATP anymore. You're just going to be producing what you can through glycolysis. And we'll learn more about the specific type of fermentation that occurs when your cells don't have that oxygen. Okay, and we mentioned it as lactic acid fermentation. We'll discuss it more here next week. So oxygen's there to accept it, drives the formation of an H plus gradient in the intermembrane space. A bunch of H plus up in the intermembrane space is able to go through ATP synthase. When it does, ATP synthase is kind of like a rotor, or think of it as a water wheel. As the H plus ions move through ATP synthase, it spins it, and that spinning creates the energy necessary to take ADP plus P and form it into ATP. And then you have your formation of 26 to 28 ATP, or in total about 32. Thank you guys for taking the time to listen to this today. I will be back on Monday, and we will go over this more and make sure you feel good for our test. Enjoy your weekend.